So the third experiment, which we planned to do with you, and unfortunately you couldn't see, you have seen only part of it, is the LAC-Z staining for detection of regions, organs, cells, and timing of expression of the gene of interest. So in our case, it is TIS7, which we cloned with a LAC-Z neocassette in order to generate a knock-in construct. That means that everywhere where TIS7 gene would be expressed, at that time when we isolate the organs or tissues or embryos, there is a possibility to detect blue staining as a documentation of the activity of beta-galactosidase. So what you have seen was an adult organism where we have isolated different organs and embedded them in tissue tag. This is for shock freezing technique the best approach because the quality of the material is protected well, the structure, and that is also suitable for proper cutting, sectioning, and afterwards for proper labeling. There are also other possibilities how to embed the material. You can first fix and then embed it and then stain it. This is the opposite case. This depends always on the on the quality of the material, on the type of organs and so on. So it has to be tested first, and the technique has to be established for each purpose individually. So what you have seen was the embedding of organs, and now I will proceed with the next step, which is the sectioning. So this is done in an instrument called cryostat, and you will see that soon. So here you see this cryostat, which is a machine where the temperature is -20 degrees. So everything where we work with this embedded material is cooled down. And what you see on at the beginning of this video are our embedded materials, which were in tissue tag, frozen and stored. First step always is that you remove the block with this tissue tag with your organ, and you have to fix it using this tissue tag on an aluminum holder at -20 degrees. One important trick is the way how you embed your organs, because this is then affecting the position of your material in the block and thereby also cutting of the organ. So here is another one. This one is in preparation. This one was already frozen to this holder. Then the holder was put into this part of the instrument. And this part is the knife, which is a hard material, which is very sharp on this edge. And with this knife is then cut the section from the block. Block is shown here. And then here you can adjust the thickness of your sections. So usually these are semi-thin, which means 7 micrometers thick sections. And you have to go through several sections which will bring you to that part of the organ which is interesting for the staining. And then you will see the attempt to cut an appropriate thin section. This is rather tricky. It has to be exercised a lot until you are able to do it. Then this thin section, which should have no breakage of the tissue is attached to the microscopic slide, and that you can see nicely here. So this is our organ to be stained, and this is the tissue tag, and it is whole attached to the microscope slide. Then the next step is the staining. So the composition of buffers you have in the protocol, which I send you via email for your information. And there you have, on one hand, fixation buffer, which contains glutaraldehyde, and then staining buffer, which is important for the staining of the LAC-Z activity in the section. Steps which were tested, I showed you in my presentation various options which can be used, and what we have established for this particular staining in this type of mice is shown here. So fixation for 3 hours, washing with the washing buffer, which was shown above, staining overnight. So this is staining of sections. It can be used also for whole mount embryos, but there, as you know already, the problem is with the background, or on the other hand, the problem can be also that the whole embryo is not completely stained if it's a thicker part of the organism where the stain does not get through. So this has certain disadvantages. Then this protocol on the top is actually for the whole mount. So the cutting is afterwards. First it's stained and then it's cut. What you will see and what partially you have seen already was that organs were embedded, and then we go through the sectioning, which you have seen now. Then these sections have to be fixed because this was not fixed yet with the fixation solution on the microscopic slides that you have seen before. Everything has to be properly labeled. Then here around this marking is used a special pen, which is a wax pen, so it does not allow the solutions to get out from this circle, and thereby your sample will not dry out, but it will be in contact with the solution. So if everything goes well, it stays within this circle. If it's not perfectly done, then part of the solution will get lost, which is not optimal. Then it will be fixed for 15 minutes. And then we come to the next step, which is the washing step. Using vacuum pump, you suck away this solution, and then you add the washing solution several times in order to allow good staining. So this washing solution is added on the material itself, on these sections, 3 times 5 minutes. And then the staining is performed overnight. So important is the exchange of this washing buffer several times. And then staining is overnight at room temperature. So sections are already fixed. Then they are kept at room temperature in dark, in so-called humidified chamber, which is a very simple device. Here is the staining solution composition you have in the protocol. So once again, you have to get rid of the washing buffer. And you put on the staining solution. Here you see it's in this so-called humidified chamber. So this is a filter paper which is soaked in PBS. Then all samples have to be properly covered with the staining solution and closed in dark overnight at room temperature. And here are examples of the stainings. So you have access to all samples in Dropbox. I sent to you all successful stainings which were analyzed by microscopy, different magnification. So in this case, it's 40 times magnification, and there you see also the size bar. And then your goal is to figure out what is what in this image. You see very successful staining. Why successful? Because there is not too much background and there are specific stainings of certain structures. What is it? You have to figure out. Naturally, it's a difficult question because you don't have so-called counter staining. So you don't see the structure of the tissue of the cells. That you have to search a bit. In Atlas, you can find images in histological atlas or in internet. What I can tell you is only that this section is from the heart. And so try to get as much information as possible, what you would do also under normal conditions in the lab when you want to characterize your sample. So I have put here a couple of images, all of them you have in the Dropbox. So jejunum, I tried to explain you what we know about the function of this gene and its gene product, protein Tis7, in the epithelium of digestive tract. And here you see quite well the typical structure of the small intestines. The way of sectioning you can identify because you can see these structures here, and then you can see also structures here, and then you see also this. So if you try to search a bit in the histology of small intestines images in the histological atlas, you will be certainly able to characterize this structure, and this one. And then you can write a comment in your protocol, what is stained, what is positive for LAG-Z and what not. Then we go to the next sample, which is skeletal muscle. And here, what I expect from you is, just a second, I have to go back. It's actually a very nice image. So sorry, we have to go. This is once again jejunum, but I will try to stop it. Okay, here, skeletal muscles. So what is very typical, how you can recognize that it's muscle, is this typical pattern of staining, which would tell you what is this tissue, because this is very typical for skeletal muscle. The reason for that you should find yourself. Then another question is, in which way was performed this sectioning? So you see these structures here. They are very typical for skeletal muscles. So the question is it a cross section? Is it the length section? And the next question is, what are these stained structures which are strongly bluish and they have typical localization? So try to figure out that and write it in your comments to this image. And then I have put into this presentation also this one, which is staining of brain. And as far as we know, the expression of T7 should be relatively weak in the brain, but still you see nice specific stainings here. So try to think about the options, what may be stained. Unfortunately, here is no counter staining, so the structure is not easy to identify from which part of the brain is this section made and so on. But in theory, with a little bit more search, you should be able to figure out which type of cells may be stained for this 7 in the brain. So what I want to say as a summary of this experiment, that it was successful because the staining was very nice and it was visible after overnight incubation with the dye. So it's quite strong staining. Another advantage is that we don't have too much background. So also the washing steps were well done and fixation was well done, because in some of these examples which I have shown you, you can nicely see the structure of the tissue of the organ, the typical staining. So that means that the quality of the material was very good. Are there any questions?
Yes, I have a question.
Okay.
In the LAC-Z staining protocol, there was stated something of X-gal staining of embryos, but I thought we didn't stain organs of an embryo, but of an adult mouse.
No, because there are two options. So either you can stain the whole embryo and then do the sectioning, or is performed this type of experiment staining as I have shown you here, which is first embedding, then sectioning, then staining, and this is used for organs. Also, naturally, for bigger embryos, it can be used. The reason why you don't see in this video an embryo, which we did before, is that now we are, first of all, trying not to kill unnecessary animals. Secondary, it's very expensive keeping breeding of animals and so on. So for purpose of an exercise in this practical course, we are not killing animals anymore, not isolating embryos and staining them, but the other way around, we are trying to use mice which would not be used for any other experiment. So this is an older animal which would be selected out from our breeding and was used for this purpose.
So in the protocol, we describe the staining of the organs of the adult mice, but you send us the protocol of the embryos because we should know the buffers.
Exactly. If you want to use this technique in the future, it's useful to have this protocol, because that is the working protocol which can be used if you are starting a new project, when you are starting to work with a new genetically modified mouse model and you want to characterize embryos.
Okay, but in the protocol we describe it with the adult mouse.
Exactly.
Yeah, okay,
so
thank you.
What I expect, actually, that's a good question, because what I expect from you is not describing the protocol as such. You don't need to do that. What I expect from you is that you understand how was this model generated. That means the cloning of the construct, what is there, which I explained in the lecture, and you also got the literature for that. And then you explain in principle the staining technique, how it is used, and you try to describe the examples of microscopic images which I sent you.